Q-Linea with the CEO Stuart Gander. You're welcome to take it away.
Great, thank you so much, Philip, and really appreciate everyone who's stayed on towards the end of the agenda here. I know it's getting later in the afternoon. I'm joining you from the other side of the Atlantic. I'm based out of Boston myself. Maybe a couple words on myself as I'm newly joined to the Q-Linea team here. I started in the CEO role at the beginning of March, so I'm roughly 6 weeks or so in on that, taking over from the founder and my predecessor in role, Jonas Jarvius. Born in Canada, I'm Canadian, but I've had the privilege of living in Sweden for around 14 years. The kids are all born there, and I will be moving back there next summer as well, so looking forward to that. What I plan to do is just spend maybe about 15 minutes introducing Q-Linea for those who are less familiar with our technology and our area of focus, and then I'll be open for questions after that. Maybe just a quick flash here. Obviously, any forward-looking statements and everything covered with the disclaimer, I'll assume this is read and understood. So a few points for today that I really would like everyone to take away from this one. So number one, Q-Linea is the leader in a field called antimicrobial susceptibility testing. Specifically, we have a diagnostic instrument that allows you to tell what drug will work at what level of concentration to address appropriately a septic or blood infection. This solution addresses a very real and urgent need across all global healthcare systems. It's a major driver of mortality in even the rich world, as well as globally, and also has huge cost implications for healthcare systems in managing sepsis. I'll talk more about that in detail. Thirdly, our solution, the ASTar platform, delivers a uniquely fast, accurate, comprehensive, and easy to use diagnostic result for the microbiology lab that can be communicated to clinicians who can thereby adjust patient therapy. And finally, right now, we're at a really interesting inflection point in the company. We have started our commercialization process in Europe already. I will share a little bit of the results there from our initial tender wins and our outlook. We are also very eager and shortly anticipating news from the FDA on our US clearance, which will allow us to go full steam ahead on our commercialization plans in the US. So I'll speak more to that in detail as well. So with that introduction, I'll deep dive a little bit on each of those points in more detail. So first of all, our overall mission, we are addressing, as I said, one of the major causes of mortality in the modern world, namely sepsis, which kills more than 500,000 people every year in the US and Europe alone. The primary cause for this is that sepsis is a escalating and cascading disease situation where if you do not address it very early on with appropriate therapeutics, it can lead to a stage where the patient can no longer be treated appropriately and thus result in death. So roughly half of all patients who die could conceivably be addressed more readily and more appropriately if we knew what type of drug in what concentration would do so. Managing sepsis is hugely costly. Patients are often in the ICU, the most expensive part of the hospital, for very long periods of time. And sepsis is, in fact, the #1 driver of cost in the US healthcare system and broadly in healthcare systems around the world. Further to sepsis specifically, we also have an underlying and increasingly urgent challenge in our modern society, which is antimicrobial resistance. So as we apply more and more therapeutics to address and combat the increased resistance from microbes, there is a ongoing war, let's say, against those bacteria. They become more resistant, and we develop and launch more drugs. At some point, though, this runs out. We are running short of therapeutic solutions to address the most aggressive of bacteria. In general, our solution can help reduce this by making sure that we use the appropriate therapeutic at the appropriate concentration and therefore reduce the amount of resistance development in a bacteria and in a population at large. And over time, this will reduce the overall antimicrobial resistance challenge. So this is our mission. We're extremely excited and committed to bringing improved therapeutics to the market. Just to give a sense of what happens today. So obviously sepsis today is addressed and treated. The challenge is really one of time. The current workflow in the laboratory right now is that once a patient has demonstrated some initial signs and symptoms of potentially being septic, a blood draw will be taken, and this blood draw will be incubated until such point where an alarm will go off indicating that it is positive for some bacterial infection. At that point, really the diagnostic workflow starts. The first step is to take a gram stain to broadly indicate what family of bacteria we're dealing with, and then when the time is ready, to run a ID on that bacteria so you can get a very specific type of bacteria so you know what you're dealing with. In a normal workflow, there's a number of other additional steps that result from this point on, where you are culturing and developing the bacterial colony in order to determine what type of drug will work at what concentration. And this time can take anything from 24 hours up to 72 hours. So by the time you have a final result that can really indicate an appropriate therapy, you might be on day 3 or day 4. Challenge with sepsis is that everything after 12 hours of that positive alarm for blood infection, you're now really increasing the risk of cascading septic shock and very negative patient outcomes. What our solution does is really accelerate that final series of steps. So at the same time, in parallel as you're doing the rapid ID, you can start our rapid AST diagnostic, and within around 6 hours, you will have a full result, which you can present to the physician to suggest what the optimum therapy would be. So it turns that between 40 and 70-hour workflow into a 6-hour workflow with considerably fewer steps. How do we do it, and what's the platform? So our main product here to address this is called the ASTar instrument. It is, as a diagnostic instrument, extremely easy to use. So it's been designed with the lab technician in mind, and to only have the most 2 minutes of hands-on time, basically a very simple pipetting exercise to put a blood sample into the cartridge, put the cartridge into a shelf in the instrument, a CD-shaped diagnostic panel into the instrument to go along with it, and you basically hit go. At that point, the workflow is completely automated, and within 6 hours, as I said, you get the result. The system has random access and continuous access, meaning at any time a patient sample comes into the lab, you can put it into the instrument. It doesn't disrupt any ongoing tests that are being done. This is critical, as obviously with septic patients, you can't plan them and you cannot batch these patients, at least not without negative consequences of taking time to get that clinical result. The platform itself can handle up to 12 samples in parallel, which is more than sufficient for a large clinical lab. I'll speak a little bit more to the panel here, but suffice to say that it's a very large antibiotic panel with a wide range of therapeutic drugs, and also the bugs against which those drugs act. Specifically for our platform, we also provide very wide concentration ranges, which enables a very granular result for the physician to know specifically what concentration to give the patient. You obviously always want to give enough drug to combat the bacterial infection, but more drug obviously drives some cost, but more importantly, it starts to drive unneeded toxicological effects on the patient, which can lead to further harm and organ damage, etc. So in initial evaluations and deployments of ASTar that we've had in Europe, we have seen that up to 95% of patients are receiving a clear result from their ASTar test, which is fantastic in terms of a physician being able to rely on getting something back once they've initiated this test. We're not the only ones in this space that have identified and are keen to address the severe challenges of sepsis. So maybe a few words just on our competitive environment and how we see ourselves fitting in. So first of all, we would make a distinction right off the bat between what we call fully automatic and semi-automatic, semi-automated platforms. Those that are fully automatic, but basically plug and play. So you put in a cartridge and or a number of consumables, and you walk away and come back with the result. Semi-automatic, you need to be hands-on in the lab with a number of the steps, which obviously drives labor and more complexity for the lab to manage. With respect to the automated systems, we are the easiest. So we have the fewest number of reagents and components. And as I said, it's really less than 2 minutes of hands-on time, which is extraordinarily easy. In addition to that, we're the only truly random access and continuous access platform that's out there, and are able to drive significant throughput with that. I would say finally also, our platform is able to handle both fastidious and non-fastidious, which in addition to the gram-positive and gram-negative is another way of categorizing the families of bacteria. And specifically the fastidious ones are particularly challenging to manage in this environment, and our platform is able to do that. So in total, what that results is we have the most data points, as I mentioned earlier, in terms of those combinations of drug-bug and the MIC concentrations to address them. And that really puts us as a standout in terms of what the diagnostic richness of the information that's being given to the physician. This is just an overview of our panel that's approved in the EU. So as I said, we've started already to commercialize this. I'll talk more about the tender responses, initial customer feedback that we're getting, but suffice to say it's very positive. We really address the breadth of the major infectious drivers in Europe, and also those small but very nasty ones, and a very wide range of drugs and the concentration. So there's a little image here that you can see in the center of the screen, which shows that CD type disk, which is a very elegant solution in terms of the ability to deliver a lot of individual data points that are represented by each of the wells on the disk, essentially where the bacteria grows. And of course, there are increasing levels of concentration of the drug in each well. So at some point, the bacteria is not growing, and that is the level of concentration at which the result is given to combat the bacteria. The point here is that on this disk, we are able to place 336 individual wells. And this compares to what most of our competitors would have, which is a standard 96 well plate. So you could say, 3.5 times more data points for any given test that can be run. This allows both more richness in the immediate term, but also over time allows us much more flexibility to add drugs and drug-bug combinations, as we will continually be getting new drugs into the market as resistance grows. So a few words on the market overall. I spoke to the very high and urgent need here. And in our immediate priority markets, which is essentially the US and Western Europe, overall, there are roughly 20 million positive blood cultures. There are a lot more of these taken, but fortunately, most of the blood draws are negative. Roughly 10% in a typical clinical environment will come back positive. That can be between 7 and 12%, up to 15%, depending on the clinical environment. We would say of those, a subset initially will likely be addressed with rapid AST. In general, our view is, and clinically would be beneficial for all blood samples that are positive to be run with rapid AST. But initially, we think that labs and clinicians will triage this somewhat. So we've anticipated that 20-25% of these would be immediately and highly urgently addressable with rapid AST. So leaving something in the order of 4 million tests in our addressed markets. And then just, obviously, if we take a roughly even cut amongst the players out there, still leaves a very attractive and sizable market for us. We would contend that we would expect a disproportionate share of that market, given the very compelling nature of our solution for this market. Sepsis isn't the only cause of infection, clearly, and not even the only major and serious one to address. There are additional drivers of mortality and morbidity, and for example, lung infection, very complex urinary tract infections, which can lead to kidney complications, etc. That also would be of high interest for rapid AST, and we have a solution designed explicitly to be able to handle these different testing formats, whether it's sputum or urine, and the instrument is ready, and we will be aiming to develop the panels for these over time. In addition to that, the bulk right now of testing takes place on isolates, and this is a type of test that we can run and would look to expand with over time. At present, that is done in a very routine basis at a much lower cost base, so it's not our immediate priority. But as costs come down for rapid AST and more urgency and need is seen in infection management in general, we expect this to tip over. So this is an area that we're quite interested in. In fact, initial discussions with customers have indicated that there are several major ones who would already start to like to experiment with rapid AST in an isolated environment, which is very positive. I've referred to on several times in terms of customer evaluation and customer feedback. So we've had a chance to deploy in more than a dozen places in Europe already, with university very consistent, very positive feedback about really around those lines that I spoke to. So in terms of that ease of use, which is critical for uptake of a new diagnostic tool, it really fits into the lab workflow in a very easy environment. It takes up a relatively small amount of footprint on the bench, which in a very crowded lab environment, as some of you may have seen, this is really imperative. Also, there's that richness of the data that's provided makes it highly clinically relevant as well. So the feedback has been really universally positive, and we have a number of ongoing evaluations, and specifically a couple trials right now in both Italy and Belgium that have been delivering very positive results. So we made a press release a little while earlier with some results presented at the AMPLI conference in May in Italy, where the evidence is showing that up to 45% of patients are receiving a result that can impact, or that is impacting, the clinical decision on therapy, which is positive both in cases where that therapy is being escalated, meaning the patient is getting or therapy changed, escalated change, which means therapy is that was being given, empiric therapy, would not necessarily have fully addressed the infection. So for these patients, obviously that new result is very timely and critical for their outcomes. But also in roughly a third of the cases, these are being de-escalated, which means that the patient is not receiving maybe the intensity of drug treatment that might otherwise have been given, and thereby saving them some impacts from the therapy. These results are being given, as we've shown in the studies, up to 20 to 34 hours earlier than the current standard of care, which is really fantastic to see. Talk about that 12-hour window. I mean, this is very significant in terms of the amount of time to really getting an appropriate therapy to the patient. And so this is translating into very positive initial discussions commercially. Before I joined the company, appropriately started those commercial discussions in Italy, where the antimicrobial resistance environment is an acute one. It is a growing one across Europe and the US in general, but some countries in specific have more challenge, and we are focusing our attentions initially on these ones for the positive impact and the general awareness, and have invested in driving that awareness, both of the need, which is generally well understood in Italy by clinicians in the space, but also specifically of the availability of new technologies to address this. So we're really pioneering the rapid AST space here. We've been developing a very good pipeline of tenders that are coming. Generally in Southern Europe, we see those as tender-driven processes, and we were happy to announce our first tender win, which is now shipped to Italy and is in clinical use now. And maybe just a few highlights from that tender outcome, which I think is helpful in illustrating how we think this will generally play out as we go forward. So the tender value was capped at around 600,000 euros. So we knew we couldn't come in over that, and was split between the financial scorings, basically our price, and the technical score, which was a range of different requirements and features that they were looking for from the different competitors. And that split was roughly 30-70. On the financial score, we came in just under the tender cap. We were very confident on the technical nature of our platform. It is truly differentiating from the others. I'll speak to that in a second. So we received the lowest score on the financial side of the players who were allowed through. On the technical side, we really had the full range, and were able to address all of the needs. So we achieved a clear premium result versus the competitors on the technical side. Notably, one of the competitors didn't pass some of the threshold criteria that were required for going through to the final phase, so we're awarded a 0. We think over time some of these key threshold criteria are going to be important in terms of that usability and clinical applicability of the results, and we are obviously very favorably positioned, as I spoke to before. So with all of that, the scoring came out quite clearly with us in favor, and we were awarded the contract. I would comment that in general, in our conversations with customers, we're seeing very consistent criteria themselves, sometimes different weighting between the criteria in terms of how the customers think about their priorities. But overall, that set of requirements is very similar across, and really in line with what I spoke to earlier in terms of the key features of the ASTar platform. So we're full speed already in Europe. We have a direct sales team in Italy, which has been building up a very good pipeline of conversations and tenders. So we're hoping to be able to announce some more wins soon. And we have direct engagement also into the Nordic market. More broadly in Europe, in order to give us some immediate scale and reach, given we're a small company with a novel platform, we've made a decision to go with distributors across France, the UK, the Benelux area, and helping us reach into Eastern Europe as well. And those pipelines are also building nicely through those distributors. A little bit of lead time, obviously, since they need we need to educate them, they need to get into their market and educate. So there's kind of two-step process, but we see those pipelines starting to grow now as well. And as I mentioned at the very beginning of my talk here, we're very excited about the US FDA clearance, which we hope to be able to announce imminently, really. We have started appropriate pre-market activities in the US just to engage with customers, let them know we're here, let them know about Rapid AST. The US market is in general more familiar with Rapid AST since one of the players was launched earlier in the US. So they're open to and very receptive to solution. I would say that initial launch was more of a Gen 1 type platform that didn't quite meet all of the needs of most customers. And therefore the market's really enthusiastically open for a Gen 2 or next gen type offering, which we have ready as soon as we get that clearance. In addition to general conversations in this regard, we've also got a couple early access programs where the platform is operational in the lab, not giving out clinical results for patients yet, but they're able to compare in the lab the workflow and obviously the results to what they're getting from the current standard of care. And the results communicated back from these partners have been extremely positive. So they were very keen to pursue this further as soon as we have that clearance. So we'll certainly be announcing that as soon as possible. But with that said, we're ramping up our commercial activities and looking forward to the next stage of top line growth. Maybe one final comment. As I said, we're not stopping at sepsis. We've got a number of innovations in the pipeline. So for the next few years, at least, we're a very full development pipeline. The company also has some very novel technologies protected with IP that will allow us to go into more point of care environment, which is useful for example, urine testing, general urine testing, or a more robust and comprehensive combination of identification and AST results direct from whole blood in our Astrid platform. The time for the market is not there just yet, but those technologies are in place and really concurrent with the ASTAR platform in many regards. So that will be sort of a future generation of product development. With that, maybe I'll turn it over to some questions.
Thank you, Stuart, for that presentation. And speaking about the public tender in Italy that you won, do you expect competitors to maybe lower prices to compete with you?
So there's a challenge, I think, in general with everyone just lowering prices, since there's obviously the cost of goods to develop these is material. Our consumable solution is also relatively simple compared to some of the competitors with the fully automated solution. So we think that we're actually quite favorably positioned from a cost perspective over time. Obviously, we're at very low scale and volume right now, as is everyone. So hard to compare like to like, but we can see the engineering and components involved. So we think over time we are very well positioned on the automated side. For the semi-automated platforms, it's true they're able to cut out a number of costs since it's a much different offering and it's a lower cost instrument. That said, labor is a real premium in a lab, as is space, and those semi-automated platforms can take more space as well and are more complicated to run in terms of workflow steps. So yes, there will be, and we can see from the tenders in terms of the split between semi-automated and automated, there's a clear distinction. We think that's a very manageable trade-off and communication with customers, and we were pleased to see that the competitors were around us on the pricing as well. This will continue to play out competitively over time, obviously, as they try to find the winning price for them versus a best-in-class offering that we have. But again, as long as we can continue to grow and get the scale, I'm quite confident we can match them on any pricing components.
And do you think incentives from above, from authorities, is necessary to generate substantial sales in the future?
When you say incentives from authorities in terms of reimbursement, or?
Yeah, in terms of lowering the risk of multi-resistant bacteria.
Yeah, I think there's certainly a benefit in terms of general awareness and pressure on healthcare systems. So we see that in some areas. For example, Italy, the UK is also a market that's had a high level of awareness and some top-down initiatives driven by the NHS, which are really good to see. I would say they open the door. They don't necessarily solve the funding challenges in labs, because labs still need to find the budgets, but they certainly put it on the priority list so a lab can feel confident and go in terms of going ahead and putting this on their procurement planning basis. I would say the main limitation right now is just getting those budgets established in Europe. There's typically a 1 to 2 year lead time to get the budgets ready and the buying, therefore, and the tenders set up behind that. And we've started to see that market. So we should be seeing the fruits of that over time. In the US, we have a breakthrough device designation already from the FDA, which is great to see. That makes us eligible for CMS's new technology add-on payment, which would fund up to 2/3 of the cost of the service for a lab, which would be a huge benefit, obviously. We can't communicate that we have that yet. We won't until we'll have clearance. But obviously, should we be awarded that NTAP funding as well, that will facilitate more rapid adoption in the US.
Time moves fast, Stuart, but thank you for the presentation and good luck ahead.
Thank you so much, Philip. Yeah, and thank you everyone for staying on the call. Really appreciate that.